Hello, grade 9 students. In this video, we're going to make a quick revision about digestive system. Chapter 1, Activity 1, our food. In this chapter, we're going to talk about the digestive system. What do we eat? What are the different forms of food? And what are the final results? What happens to the food inside our body? To start first, let's talk about digestion. Digestion is the transformation of large complex molecules into simplest one to be used by the body cells to produce the energy needed. So which system is responsible? Sure, it's the digestive system. It's divided into digestive tube or digestive glands. Digestive tube, they are organs through which the food pass, whereas digestive glands, they are organs through which food doesn't. Okay? Organs of the digestive tube include the mouth, esophagus, the stomach, small intestine, large intestine, and finally the anus. Digestive glands include gastric glands, salivary glands, liver, pancreas, and intestinal glands. You have to know that digestive glands, what do? Digestive glands, they secrete digestive juices into their site of action. And also you have to know that the the first they are divided into carbohydrates, where the complex form is called polysaccharide, intermediate is called disaccharide, made up of two simple sugars, and the simplest form is called the monosaccharides. Proteins, they are composed complex forms called proteins, more than 52 amino acid chains, polypeptides, less than 52, and amino acids, they are made of only one pair. Lipids, they are very energetic. Complex form is called the lipids, and the simplest form is called fatty acid and glycerol. There is no simplest, there is no intermediate form. You have to know that the water test is done to identify the water. Salt test is done to identify the presence of salt, where we have a positive result as white precipitate. Fehmi test is done to identify the presence of reducing sugar, where it gives brick red precipitate. Iodine test is done to identify the presence of starch, where it gives a dark blue color, and violet test, it gives a violet color whenever it's added to proteins and or polypeptides. Coagulation test is done to identify the presence of only proteins. If the food coagulates, so this is protein. If it doesn't, so it's not a protein. Translucent spot test is done to identify the presence of lipids, where translucent spot is formed. And also we have Southern test is done to define the lipids where pink color appear. So you have to know that digestive juices, all of them, saliva, the pancreatic juice, intestinal juices, gastric juice, all of them they contain enzymes, except bile. So what are enzymes? Enzymes, they are the biological scissors that digest or break down food into smaller pieces, and they remain unchanged at the end of the reaction. You have to know that the amount we have saliva that contains salivary amylase, it works on starch and kills maltose, where the medium is neutral, meaning that the pH is equal to 7. Stomach, gastric glands secrete gastric juice that contain the enzyme pepsin that works on protein to get peptide. Also, you have to know that pancreas secretes pancreatic juice that contains the enzyme peptidase, works on peptides to give amino acid. Also, trypsin, it works on protein to give amino acid. Amylase, it works on starch to give the maltose, the medium is basic. Lipase works on lipids to give fatty acid and glycerol. Bile doesn't contain enzyme. It works on lipids. It makes emulsification of the lipids. It dissolves or it dissociates the lipids for helping the enzyme lipase to digest them. Intestinal glands, they secrete intestinal juice. Repsin works on peptides to give protein. Maltase, it works on maltose. Lactase, it works on lactose. And lipase works on lipids also. So you have to review the notes regarding the enzymes, their substrates, and the final results, as well as the chemical medium. Moving to chapter 1, activity 2, chemical transformation of food. You have to know these two tubes A and B, both of them contain corticosteroids, while tube A contains small amount of fresh saliva. Saliva contains an enzyme called salivary amylase. It works on starch. So digestion in tube A and B at time equals 0 minutes, iodine test, it gives positive result, dark blue, while after 15 minutes, Tube B remains dark blue, meaning that it still has starch, while tube A, there is no dark blue. The starch, it's transformed into another substance. While with final test solution, you have time zero, both of them they give dark blue, meaning that it's a negative result. And the positive is Greek red precipitate. In tube A, we have Greek red precipitate, meaning that the starch is transformed into a reducing sugar, which is a maltase. So tube A is a control tube. The substrate here is called a starch. The variable factor is the presence of saliva. Aim to study the effect of saliva. Pose a problem is similar to the aim, but it should be form of question. So what is the role of saliva? Table of conditions you have to go with. What are they contain? Substrate, saliva, water bath, temperature, time, and if they want the result, you have to add the result. The legend plus means presence and minus means absence. Moving to chapter 1, activity 3, the enzymes agents of digestion. So you have to know that first we have two tubes, A and B. One of them contains protein starch, and the second one contains albumin of an egg, which is a protein. So tube A contains fresh saliva, while tube B contains also fresh saliva. Tube A, after about 1 hour, it gives with final test regret precipitate. So the starch is digested into reducing sugar, whereas in tube B, albumin of an egg is not digested since it gives a direct test, gives a direct color, so we have protein. So fresh saliva, it has an enzyme called salivary amylase. Salivary amylase, it works on starch and not on protein. So the starch is digested into the reducing sugar, whereas the protein is not digested into amino acids or is not digested at all by the enzyme salivary amylase. So the first property that can be used here that the enzyme acts on a specific substrate. Every enzyme it works on a specific substrate. And in this case, saliva, enzyme in the saliva is called salivary amylase. It works on starch and digests it into reducing sugar, but it cannot digest the albumin. Moving to this figure showing the different pH medium for three different enzymes, pepsin, salivary amylase, and trypsin. Pepsin, as you know, pepsin as Pepsi, it's acid. So it works in acidic medium at pH equal 2 to 2.5. It works at its best. As the temperature increases from 2.5, the pH increases, so the activity or the speed of reaction will decrease. So it works at best at this pH. Whereas the enzyme salivary amylase work at best at 6.8, approximately 7. So it works in a neutral medium. Whereas the enzyme trypsin works at its best at pH equal 8 to 8.5, while as it increases to about 10, the activity or the speed of the reaction decreases to 0. So here we can deduce that trypsin increasing the pH stimulates increase the speed of reaction up to a certain limit, meaning that pH is acidic. So pepsin works in acidic medium, whereas salivary amylase it works best at pH approximately neutral, around 6.5 to 7. So this means that the enzyme of salivary amylase works in a neutral medium, whereas trypsin enzyme works at its best at 6 to 8.5, decreases to the maximum. So pH equal 8.5 is the best one. As it increases from 8.5 to 10, its speed decreases. So trypsin works in a basic medium. So the second enzymatic property, every enzyme needs a proper pH medium. It needs a proper chemical medium in order to act and work. So pepsin needs an acidic medium, salivary amylase needs a neutral, trypsin needs a basic medium. Let's move to the temperature. As you see here, at the temperature it increases from 0 to 37, the activity of the enzyme increases to the maximum to 100% at 37, since it's the normal body temperature and the enzyme works at their best at this temperature. As temperature increases from 37 to about 60, the activity of the enzyme it decreases to about 0%. 60 represents the denaturation temperature. So here, as the activity temperature increases from 0 to 37, the activity increases from 0 to 100. And as temperature increases from 37 to 60, this activity decreases back to 0. This indicates that increase in the temperature stimulates an increase in the activity of an enzyme up to a certain limit, which is 37. So here, every enzyme needs a proper temperature to act. So till now, we have three characteristics or three properties for the enzyme. Number one, every enzyme works on a specific substrate. Number two, every enzyme works in proper pH medium, whether acidic, basic, or neutral. And every enzyme works at proper temperature, which is about 37 degrees Celsius. So you have to keep in mind that optimal temperature in this case study is 37. Why? Because the enzymatic activity is at its best at this temperature. Inactive, it's 0. Why? Because the enzymes they are inactive, they are sleeping. So these enzymes they are sleeping at temperature 0, so we call it inactive temperature. 60 degrees and above, it's called the denaturation temperature, or it's called the destruction, where the enzymes they are denatured and destroyed under very high temperature. So this is what we call it denaturation temperature. So the activity will be 0. So you have to keep in mind, if the enzyme was inactive at cold temperature, then it's placed back at 37. So this enzyme will work again, will function again. While if the enzyme is placed at a very high temperature, 60 degrees and above, it will be denatured or destroyed. Even if you place them at 37 degrees, so the enzyme will be denatured and there is no more functioning of this enzyme since it's denatured at all. Chapter 1, Activity 4, Transport to Nutrients. You have to know that mechanical digestion and chemical digestion will form the parts of digestion. Mechanical digestion, it breaks down food into smaller pieces, whereas it keeps the food moving through the long digestive tube. Mechanical digestion includes different movements. In the mouth, we have chewing. In the stomach, we have churning. And in the small intestine, we have peristaltic movement where the food is going to be digested. So these represent the different actions of the mechanical digestion. Moving to the chemical involve digestion of food by the help of the juices and their enzymes like gastric juice, pancreatic juice, saliva, intestinal juice, including the enzyme salivary amylase. Gastric juice include the enzyme pepsin, trypsin, epsin, peptidase, lipase, sucrase, maltase, and others. They break down the food into simpler nutrients and they occur mainly inside the mouth, inside the stomach, and the small intestine. How are these two types of digestion needed? You have to know that mechanical digestion, it facilitates the chemical and allows the digestion to finish faster than usual. So cutting the food into smaller pieces will help the digestion finish faster and will help the action of the enzymes as well action of the juices on the food to digest it faster and helps in the process of digestion. Moving to the root of the nutrients, you have to know that there are four important characteristics. We have to study these characteristics. Four important characteristics: the fever, the intestinal absorption. Number one, we have thin wall of intestinal cells. The intestinal cells, we have thin wall, large surface of exchange, highly vascularized, it's rich in the blood vessels and lymphatic vessels. Number four, it's immature. So these four characteristics, we have to study them, the fever, the intestinal absorption. Routes of absorption, we have two different routes. Either through the blood, where it absorbs all the nutrients except fatty acid and glycerol, whereas the lymph, where it absorbs fatty acid and glycerol only. So blood characterized by absorption of all the nutrients except for the fatty acid and glycerol, whereas the lymph absorbs only fatty acid and glycerol. This is the summary for everything related to the digestive system through the first semester.